pepsin, sorry. Okay, so let's start. किसमें? 11, okay. मैं समझी एक grade आया था. Okay, which statements are correct for all enzymes? They are proteins, yes. They are secreted into alimentary canal. Uh, okay, yes. They speed up the reaction, yes. Actually, it's for all. None of them work at low pH, yes, they do. So the answer is 1 and 3. In the enzyme action, where is the active site and where is the lock and key? Yes, active site is on the enzymes. Key is the substrate. Think again. Lock is the substrate and wait. Wait, what have you said? What have you said? Enzyme is the lock. Yes, it's a. Enzyme is the lock and key is the substrate. Okay. Again, the same question is given. You have to identify the lock and key. So key is basically the substrate. So what is substrate among these and what is lock among these? Let's say key is substrate and lock is enzyme. Starch and amylase. Amylase is the enzyme that work on starch. Okay, and convert starch into maltose. Okay, wait, wait. Starch converts into maltose, maltose into sugar or glucose. So here the enzyme that work is amylase, and the enzyme that work here is maltase. Okay, basically this is amylose sugar, which is a scientific name. So we casually call or we generally call it starch. Starch is a common name. Okay. Okay. According to the lock and key hypothesis, what is the lock and the key is for enzyme? Same. Yes, the lipid and lipase. Correct. Let's move to 15th one. Sorry, I need to copy. Okay. So it's a tough one. Lots of diagrams are given and we have and their temperatures given. Wait, 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 I haven't copied yet. Okay. Here we have four beakers. First one is with 90 degrees Celsius. Second one is 35 degrees Celsius. Third one 20 degrees Celsius. And last one is 10 degrees Celsius. Okay. So we'll say this 91 is going to denature, right? Because the temperature is really high. The 31 would show the optimum activity, the maximum one, right? Sorry. 20 degree it would perform, but if ice is present, then the activity would be really, really low. Okay. Now, after 5 minutes, each test tube of amylase is poured into the test tube of starch solution in the beaker. After leaving it for 5 minutes, the sample of the mixture were tested with iodine. Okay. Now let's see the effect of iodine. What would be our answer? Let's say, let's go through the answer. So here it's broken down. So it's not going to work. So starch will present in the first one. Sorry, present. In the second one, it would not present. Starch would convert into glucose. The last one, sorry, the second last one. Yes, starch must be present along with, no, it would be denatured. It would be denatured. Okay, and the last one, because the activity is very low, more starch is present. Okay. Okay. So what changes are going to be made? So we say in 90 minutes it will remain. Why? Wait, let me. Is this for the humans? Yes, it's related to plants and animals. Okay. In 90-degree there won't be any change, right? Oh, sorry. Starch is present, so it will turn blue black. There is no change in the reaction. Starch is present, so it will turn blue black. What about 35? It is fully changed, so it will remain. No starch. There is starch in 10-degree as well, because the temperature is low. The reaction is not up to that mark that it converts the starch into glucose. This one is ice. Less temperature mean less activity. Yes, it's written starch is present. Yes, yellow brown. Okay. What about 20 degrees Celsius? Yellow brown. And what about last? That would black. So what would be your inactive? Yes. So it would have starch. It has starch, right? It will turn blue black. So our answer would be A. Sorry. Our answer would be okay. Anaya, your voice is really low. I can't hear you clearly. No, it's low. Just wait a minute. Let me check mine. I guess I need to increase the voice for my speaker. Okay. Now, now you can say. Hmm. Yes. Okay. At 35 degrees Celsius, the activity is maximum that we have said. Okay. It means that amylase is working well. What is the function of amylase? To convert starch into glucose. Okay. Okay, let's move to the next question. Cubes of hard boiled egg. Okay. Now the hard boiled egg is already denatured one. Anything that is boiled is denatured. So we cannot check the effect of enzymes or the osmosis on the boiled things. We have learned this in the movement across membrane as well. Have we? No. I think so. We haven't done the MCQs for that one. Let's discuss them today. Okay. So what will happen when you boil something? You are actually destroying the surface of or the cell membrane of that compound, whether it's plant or whether it's some other thing. Okay. So you cannot really test something which is boiled. Okay. But here the scenario is different. Here it's written the cubes of hard boiled egg whites are placed in the test tube containing 5 cubic centimeter of water. Other substances are added to each tube as shown in the chart. These tubes are left for 8 hours and then tested for amino acids. So what is the function of pepsin? Hmm. It converts protein into amino acids. So where is the positive result shown? Where there is pepsin and acid, where there is boiled pepsin and acid, and where there is an acid. Is it so? So which two which test tubes show that pepsin is an enzyme? So it's converting something, right? So it's 4 and 5. The only option that is available to us is 4 and 5. Okay. Let's move to the 17th one. Oh, these MCQs are really like taking space. They're lengthy. What are you eating now? Okay. The effect of temperature. At what temperature the enzymes get to denatured? We have discussed this. It's 50 and 60. At 60 degrees Celsius, most of the enzyme got denatured. Okay? Yes. No, its activity started from zero actually, and then decrease after a certain point, right? So that is only correct for C, not for B. Yes, but the activity is not starting from zero, it's starting from 20 in B, right? So we have choose exactly. So A and D are not in question. Sorry, these are not the optimum an enzyme cannot have so many optimum temperatures. Okay, next question. Protease break down proteins into amino acids. Again, what is log and what is key? No. Log is enzyme. ठीक है. And key is the substrate. Where's the next question? Have we discussed 18, 19th. Okay, 19th one, I guess we have discussed questions similar to it, but not this one. We discussed a similar question earlier. Oh, sorry. I need to take screenshot. Oh ho, it's really off. Okay, now okay. In an experiment to investigate the effect of heat on the germinating barley grains, three petri dishes were set up, and they are like they were different some things. One has the amylase, the second one has germinating barley grains, and the third one has the boiled germinating barley grains. Okay, okay. So in diagram number 2, after 3, the sum petri dishes viewed from above. So in the first one, we can only observe the amylase one, but in second one, the growth has been shown. In third one, there is no growth. There are only like barley grains. Okay. What is the best explanation of this one? Okay. Okay. Yes. Amylase is present only in the first one. Amylase is produced by barley grains that have been boiled. No. Amylase from barley grain is denatured when they are boiled. Okay. Germinating grains prevent iodine from staining blue-black color, and starch from the substrate is used by the grains as energy source. Okay. The experiment is related to the three different things where we are judging the germinating process, and we have added iodine. So in amylase 1, where the amylase is present only, it's not going to show, it's I guess turning blue black, or maybe lines are given. Then in second one, the what do we call it? The space around something. There is a specific word that skips out of my mind. Okay, it's getting bigger. No, no, no. It's about like where there is growth, where there is culture. It's about that. Okay, it has grows bigger, but in the third one there is no growth shown at all. So we are saying that it could be B because the barley one, in barley one, the amylase is going to be stopped. Okay. So in seeds, amylase, do you think amylase is present? In seeds, yeah, exactly. So why not A? Oh, no, no, no. Why not C, not A? Okay, B is the most appropriate one, by the way. It's a more appropriate one. B. Yeah, it's B. It's the most appropriate answer. Okay, 20th one. Enzyme action can be explained by the lock and key hypothesis. Again, what is present on the lock or key? Substrate is a lock or substrate is a key? A key. So it's A. Okay, let's move to the next ones.